All right. So as Nina mentioned, I'm going to talk about how you can query the database, how we report the data, and I'm going to go through some examples. And some of this you would have already seen with Alba and Alex's talk, but hopefully repetition is good and there'll be a little more detail as I walk through each of the ways you can query the database. So this is the homepage, as you've seen, and it has these 6 key search panels. And from these search options, more than 99% of the queries that people come to the IEDB to do can be done right here on the homepage. So these are the main ways people want to access the data. And we leave it up to you as to which of these 6, if any, are relevant to you. And starting with the first one up at the top, it's the epitope. So if you have a peptide you're interested in searching, you can enter it here, or you can do a broad search for just all linear peptides, discontinuous residues, or non-peptidic structures. We do have carbohydrates, lipids, drugs. And if you are searching on a specific peptide, keep in mind that a different author or researcher may have studied a slightly different peptide. Theirs may have been 1 or 2 amino acids longer or shorter, or there may be 1 or 2 residues different from what you enter. So if you don't get a lot of data for your specific search, consider broadening your search and using these different blast identity levels of 70, 80, 90%. You may see what other people have studied. And it's not uncommon for, if a protein isn't widely studied, for larger peptides to be searched, to be published first prior to narrowing down what precisely the epitope is. I have lost the ability to go forward. The next search pane is on the epitope source. So if the peptide is natural and it's derived from an organism, we do have unnatural peptides analogs. You can search for the organism name. And so some of the questions earlier about snakes and parasites, if you just start typing in the name, the species name of your snake, and actual common names will work too, or the parasite you're interested in, you'll get all the epitopes that we have data for. And as Alex mentioned, this reflects the literature from the beginning of PubMed until now. So we have in there, if it's been published, that there was an adaptive immune response to an epitope from your particular snake or parasite, it will be in the IEDB. So this is also useful for you to mention in your grants. If something isn't widely studied, you can justify getting money to study it. So as I mentioned, we also have the common names, and you can just start typing. And in each of these search panes, when you see the find button, it will open a tree and let you navigate and let you search on a higher level. And I'll show a few examples of clicking on the find button as I walk through the search panes. So the next one in the epitope source box is the antigen. So for peptides, this would be the protein that the epitope is derived from. But many different organisms share proteins of the same name. So you can see when I start typing capsid protein, I get a lot of capsid proteins in all the viruses. So I'll show you in my example a really quick way to navigate to a specific protein from a specific organism. You can also do this using the find button, but if you search for your organism first, you can do it with 1 click once you get to the result page. And then the next search pane is the host. So any host for which we have data, which reflects what's in the literature, can be narrowed down to using the host search pane. And we have the radio buttons for the most commonly studied hosts, but all the others we have. So we do have cow and pig and dogs and camels and all sorts of birds and fish. And so if you click on the find button, in each of these search panes, it opens a finder, which provides a tree view of all the data we have. And you can click through here and see all the mammals we have published data for, the fish, the birds. And then the next search pane, this is back up at the top of the page, is the assays. So if you want to narrow your assays right from the start on the homepage, you can do it by unchecking the ones that aren't relevant to you. You can also start typing if you know a very specific assay type you're interested in. and you can click the Find button and then see the types of assays for which we have data. And I really suggest doing this first because there's a lot of different ways to say an assay name. There's many different synonyms and abbreviations. So it might be good to see what we have, but also it may also inform your query that you may want to search on a higher level. You may want all binding data, not a specific binding assay, or all 3D structured data. So you can use the tree to search on a high level that groups assays together in a logical way. And that's, again, using the ontologies that we built into the finders. And then the next search pane is the MHC restriction. And we let you narrow on a high level of class 1, 2, or non-classical. And if you have a specific MHC molecule you're interested in, you can start typing. But again, similarly to some of the others, it's complicated to type an MHC molecule with dashes and asterisks and colons and the names. We do have all the synonyms and shorthand names, but there is a lot of similarity across species. For example, if you typed O201, you may get several different species. So if you use the tree view, you can navigate and see the types of various species for which we have MHC information. And again, this reflects the literature. So in a T-cell assay, some authors may not provide any MHC restriction information in one paper, and in another paper they may provide a precise or a vague MHC restriction. They may say it's just class 1. So we have whatever each individual publication tells us. And so here you can narrow down or go high level just with all the other trees. And then the last search pane, the disease. So this is the disease of the host at the time or before the experiment was performed. So the T cells or the antibodies were taken from a host who had a relevant disease to this specific adaptive immune response is being studied. So did they have dengue when you're studying dengue epitopes? Were they vaccinated for COVID prior to the COVID epitope experiment? And we also do capture cross-reactivity between different infectious diseases when that's what the authors were studying, or a cross-reactivity between infectious diseases and autoimmune diseases. And just like the other finders, if you can type, and it'll bring up all the possible diseases, or you can click the Find button and see the types of diseases for which we have data. So these would all be infectious diseases, allergic diseases, transplant, autoimmune, any disease that was relevant to the adaptive immune response. And clicking through gives you an idea of what we have in the database. And then I'm going to use a user query to walk through what it looks like to run the query and look at the results. And so a user had asked us, how do you see the difference between B cell and T cell responses for any pathogen? And this is also true for any allergen or any self autoantigen, or I guess parasites are pathogens. Any the snakes would count too. You would type the snake species or the common name, and the synonym will pull up the options for which we have data. So I'm doing influenza A virus for my example, and this is because we have so much data for influenza because it's so widely studied. It makes for a good example. So once you've chosen something from any of these search boxes, the homepage will show you how many options you've chosen because these are multi-select fields. You can go and select another organism if you'd like to add to it. And we don't know how many of these 6 panes you might want to search on, so you can build a query as complicated as you'd like right here on the homepage. If you only want to see human data, if you only want to see class 1 data, you can choose all of these selections here. And then once you're done and you've built your query, you hit search. But you don't have to pick anything. You can just go to the website and hit search, and then you'll see all of our data that is positive, because I want to point out the default is to have positive checked. And this is because this is what most users have shown us that they wanted. But if you want to include negative, just check it and then hit search. And the next, once you've hit search, you land on the results page. And I'm going to go through the features of how the results are presented to you. And the first thing is to show you that what you're queried will show up at the top of the page. So if you hit search and left the room and took a phone call and came back, it'll remind you what you had queried upon. And if you change your mind, you can just X these out and make a broader search. And once you've done that, you have to hit the search button again to rerun and refresh the page. So now I'm going to go through what you see on the left-hand side, because here we've provided a few more additional ways to narrow your data even further than you could on the homepage. And we provide these in a structured filter options. We have several different filter options depending on the type of researcher you are, that we can make these even more fine-tuned to your needs within the same search pane. So when you first land on the results page, if you haven't picked anything that made your query specifically T-cell, B-cell, or MHC, you'll land on the default filter options. And the default filter options gives you a set of additional filters that weren't on the home page, and they are in the Epitope search box. It looks the same as what you saw before, but now you can also search for 3D structures that have been curated. And you can search on amino acid modifications, so post-translational modifications made to peptides. And sometimes these can be really important in immune recognition. And then in the Epitope source search pane, in addition to what you had on the homepage, you now can include related structures. So these are structures that are not from influenza A virus, but are related to it. So for example, analogs. So if you want to include non-natural peptides that are similar to influenza and were studied as mutations of influenza peptides. And then we add two new search panes that aren't on the homepage, the receptor and the reference. And the receptor are TCR and BCR sequences. So if the authors provide us with nucleotide or protein or CDR1, 2, or 3 sequences, we capture whatever the authors give us in the paper for any epitope-specific TCR or BCR. And then the reference is where the data came from. And there were questions earlier about journal articles and submissions. If you want to only see journal articles, you can do that here. If you're just curious what submissions we have relevant to your query, you can look at them separately. If you do any, you'll get both back in your results. If you want to see a particular author's papers, if you have a title or a PubMed ID to narrow to a very precise type of paper, or if you want to limit to specific years, you can do all of that here. And then if you use that drop down to look at different filter options, if you're a T-cell researcher and you want to search for specifically T-cell data, we provide you with a few extra ways to narrow your results to just T-cell data. So the receptor search pane is searching specifically TCRs in this case, alpha and beta chains. The assay box is searching specifically T-cell assays, with some of the more common ones right on the home page, right on the results page as radio buttons. And then we also let you narrow to look at only direct ex vivo detection if you would like. We have a lot of data on T-cell clones and short-term re-stimulated T-cells. And if you don't want to see those and you only want to see T-cells that were taken directly out and tested without any re-stimulation, you can use this checkbox. And then the MHC restriction search pane is relevant to T-cell data. And we provide, again, whatever the authors give us on the epitopes. And some authors may say class 1, class 2 in a classical, but sometimes they have more precise MHC restriction. And so the resolution drop down lets you let you narrow your query to show only data that has some class restriction data, or 1 or 2 chains of the MHC molecule. So if you want really precise MHC restriction, the papers that show that precise data, you can narrow to those. And then also in the MHC restriction search pane, we provide evidence. We let you see how that MHC restriction was determined. Different papers determine the restriction in different ways. And some of you users may care very much about exactly how it was determined. For example, you may not want binding predictions, but you may want MHC binding assays. So we tell you how those authors asserted that evidence, asserted that MHC restriction. I'm trying to move my, I don't know what's happening there. So the next type of filter option is B cell. So if you're an antibody researcher, you're only interested in searching for antibody data. You can search across BCRs, antibodies, heavy and light chain information. And in the assay fields, you're searching only on antibody assays. And we've put radio buttons out easy to find for key types of assays that people like to search on. And then we also let you search for specific isotypes. If you want to narrow to IgE or IgA or IgG specifically, you can do that. Then the last type of filter option we have is MHC. So for MHC researcher, we separate MHC binding assays from MHC ligand elution assays. And then again, we provide in the MHC restriction search box the resolution and the evidence, because we do have MHC binding data that's on a high level of class 1 or class 2, if that's what the authors tell us. And if you want to look at only precise data, you can use the resolution field for that. And then back out to the results again. Alex had gone over this a little bit about we have the different tabs to present the data to you in different formats. Each of these tabs have slightly different columns relative to what they're showing. And for each tab, we have a header bar that has little carets that you can use to sort the columns if you would like to change the sorting. The default is with the most assays. And then also you'll see when you're on the epitopes tab, 3D, there's a 3D viewer for epitope structures. And so this displays where an individual epitope is located within its source antigen. So this will be the protein source of this epitope. And you can see where it is. And we either use models pulled from the PDB, which is what I'm showing up here, or we use AlphaFold structure database when PDB structure isn't available. And if you click on them, you can play with the controls and look at the different features. And we have help topics for all of our 3D viewers. And Lindy will be talking about the help topics later, but you can go to the help directly, or you can click on little question marks that you'll see throughout the database. And then the next thing, a key feature of all of these tabs is the ID, the first column with the IDs that are blue. When you click on them, you get more details about that row of data. So in this case, I'm on the epitopes tab. So this ID will take me to all the data about this epitope and give me more details than you can see in this table view. So click on it. I get a page that has a textual summary and a tabular summary of all of the data we've accumulated thus far. And so new data can come out every week. And so we may add new papers about this same epitope again and again as it gets studied. The beginning of the COVID pandemic, we didn't have COVID data. And at the end, we had a huge amount. So you can always come back and see how the data grows. And here's an example. This epitope is clearly an A0201 binder because we show you how often it was studied and how often it was positive, but we also show you all the experiments. There's a few HLA, class 1 HLA it was tested for that were negative, but it was only tested once. So we let you draw your own conclusions of how you would interpret this data. And also you can see what's been studied and what hasn't been studied. If it doesn't show up here, that means those binding assays haven't been published. And then you can also see that some authors provide A2 and some authors provide us with A201. And those reflect what's in those papers. And then if they're B-cell, T-cell assays, they will also show up. This is a widely studied T-cell epitope, but it does have a few B-cell assays and they were positive. And then you can also see the type of epitope it is. So interferon gamma release is highly positive, and some of these other cytokines were not studied very often, and some of them were negative more often, and some were positive more often. And then out on all of these pages, again, we have little inline filters, these little funnels, and this is where you can narrow your results right quickly on the fly. So I did a query for all influenza A epitopes. And so if you click right here and use this funnel, I will narrow my results on all the tabs very quickly to matrix protein, if that's the protein I'm interested in. So this is a way to very quickly without navigating the tree or typing matrix protein, you can narrow to it and you can be guaranteed it's from influenza virus. And then another feature that Kelly's going to talk about are exports. Each of these tabs up at the top has an export results option. And Kelly will talk you through the details about how those work and how you can change them to custom fit your needs. And then the next tab over is the antigens. And again, with the header columns. And here you click on the protein name itself. And again, also the funnel. So when you're on the antigens tab, if you've done an organism query, as I did with influenza, whatever organism you searched, you will see all the proteins for which we have data from that organism. Because influenza is so highly studied, we have data for all of its proteins. Some organisms, we may have data from only a single protein, or maybe one protein is the one that most people study, or it could be that the others just haven't been studied yet, but it gives you a good overview of what type of data is out there for that organism. So here I'm going to click on the hemagglutinin, which is blue, to let you know that something fun will happen if you click on it. And so this will show you all a summary of this protein, what we have for it. So this is the antigen summary, and we provide a textual summary of all the data we have for this protein, and we link out to UniProt. So we have lots of different hosts studied. So we have cow and duck and goat and rat and horse, crab-eating macaque. And then we also have proteins being studied in different disease contexts. So influenza and healthy, of course, were studied quite a bit, but some autoimmune diseases as well were studied for responses to influenza virus. And then there's a summary of how many discontinuous and linear epitopes we have. And we link out to the protein ontology, and we also link out to IPTMNet for post-translational modification information. And then the next tab over are the assays, and this is the meat of the data in the IEDB, the experimental assays. And this is where you can do a lot of analysis by exporting the assay results and then analyzing them to see all the different columns. I think there was a question about, sorry, I forget now. I hope it's still there, not answered. But it was about some detailed where you didn't see directly how you would filter for those variables through our search interface. And it's because I'll show you assay details and show you how many fields we have. We can't possibly make options to query all of them. But once you export the data, you'll see all those columns, and you can use them to filter and sort on your export. So we separate the data by the T cell, B cell, and MHC tabs on the top level, and that's because the columns of the summaries are slightly different for each of the assay types. And then there's the same headers and the same funnels. You can now quickly filter to your hosts, filter to a particular paper, class restriction. And then we also, in the assays tab, show you 3D assays. So if you click the checkbox in the epitope search for 3D structure assays, you'll get all of the 3D structure assays relevant to your query. So this would be the influenza 3D assays, since that's what my query was. And then you can get the IEDB assay viewer that shows you the experimentally determined 3D structures that were in these specific papers. So these papers determined a 3D structure and submitted it to PDB. So this PDB ID was submitted associated with this paper. And then, as we saw with the epitopes, the first column, the ID, takes you to the details about this row. So this is a quick summary of what we have, but we capture hundreds of fields per assays. So when you click on the ID, you get a page that I'm going to scroll through. This is the top of the page for assay details. And the first section is about the reference, where the data came from. So all of the data in the IEDB, we tell you where we got it from. So whether it's a submission or a paper, most of the data is from the literature, and we'll link out to that paper. And then the next section is about the epitope. All the data in the IEDB is epitope specific. So in this case, the epitope was a hemagglutinin one because of my query. And we tell you where in the paper we got this linear sequence from. So Alex mentioned we must have a structure. And so for peptides, the author must have provided exactly what amino acids they studied. And that was found in the material methods. Then the next section is immunization. And this particular example was a bulb seed mouse who was immunized with the epitope, but it could be a human who was vaccinated with the vaccine. It could have been a human who had an infectious disease. It could be a cow with an infectious disease. And we link out to ontology identifiers wherever we can, and you'll find these in this column, and these little blue arrows are link outs to those ontology terms within their external ontologies. And then the next section is about the assay itself. This happens to be a T-cell assay, so it's about the effector cells and antigen-presenting cells. But if it was an antibody assay, the information here would give you details about the antibody itself, its isotype, and its type. And so this was a tritium thymidine proliferation assay that was done with T cells and lymph node cells. And here again are the link outs. And the restriction in this paper was provided just to H2D class 2, so just class information. And how the authors determined it was with the cited reference. So they just told us this is known to be a H2D class 2 epitope. And that's, so we put whatever level of detail we have. And again, we link to the ontology identifiers. And in this experiment, the assay antigen was the epitope itself. The peptide was tested, but it isn't always. It could be a whole virus, a whole protein. It could be a cross-reactive peptide or an analog. And then we always tell you where in the paper we found this tritium thymidine experiment. It happened to be in figure 2. So you can go back to the paper if there's anything additional you want to look up. And then the next tab after we go through the assays is the receptors. So these are the TCR and BCR sequences. And again, we provide whatever the authors give us. So if they give us CDR3 sequences, we'll have them. They're not always paired, but if they do give both chain 1 and chain 2, we will enter it. And similar to the other tabs, there's the headers that you can sort by, and there's the IDs that you can click on and see details about that row. You can also hit the funnel filter and narrow to see all the data. It'll change across the tabs to show you all the data from just this group ID. This is CDR3 you're interested in. So this is what it looks like when you look at the details for the receptors. So we summarize all the data together. And in this case, there's quite a few 3D structures. Different papers provide different levels of gene usage. So you can see more details or less details. And they may be paired with different alpha and beta chains. And then a summary of all the peptides that it bound. And this particular receptor also bound some analogs of the same wild-type protein. And then the last tab over are the references. So these are the papers from which that data came, and it has all the same features as all of the other tabs. And then there was a query. I see Nina is back, so she may want me to stop. We wanted to talk.
Your minutes, Randy.
Okay, we wanted to talk about how you can see the difference between B and T cell responses. And so this is for any organism, not just a pathogen. And you can do it by looking at the epitopes on the different tabs, T and B. They will look different. You can export them and then compare them and do analysis that way. You can change your query to look specifically B or T using the checkboxes. You can repeat the same query one way. So this was, I took off that I want no B cell, no MHC, so just T cell. And then you can repeat it again. And now I'm saying I don't want T cell. I'm looking just for B cell. And you can see how it affects the number of epitopes, and you can export them. Also keep in mind the same epitope can be tested for both T and B responses, or T and MHC. And then it's been mentioned earlier, the Immunome browser, which is a great way to visualize the differences between responses. Both Alba and Alex talked about it, so I will go quickly. Here I just repeat my query. So whatever your query is what gets plotted on the graphs. So here I did no T cell, and here I did no B cell. So it's the B cell and T cell. And this is, in my case, my query was just for influenza A, so I didn't include a host, but you could narrow to the human response, or a bulb C response, or humans who were infected response, and you'll see differences. And you can also narrow further class 2 and class 1 responses. So here I did this using the MHC filter box. So you can build your queries however you want to make the data relevant to your specific needs. And then this is the last slide. Yeah, pretty much the last slide. I want to talk about the Immuno browser has a 3D viewer built into it also. And again, you'll see a question mark. That's where I want to point you to go to the help. There's also help here if you want to play with it. I suggest playing with it first and then reading the help articles to find all the details of how these viewers work. And then I wanted to mention that we are core trust sealed repository, and you click on that little icon and you can read more about it, but it basically tells you where you're trustworthy. Okay, Nina, that's it.
Thank you so much, Randy. That's fantastic. Hope everyone enjoyed the deep dive.